Okay. And record. Hello, everyone in the room, everyone on YouTube. Welcome back to another Water, Wetlands, and Watersheds seminar. So today we have another one of our very own from the lab. So this is Calvin Kong, Yi-Young Kong. And Calvin is a fourth-year student in the School of Natural Resources and the Environment. You can stand up here. I want to get in this picture. Thank you. So Calvin has been working in my lab since 2021. 21? Yep. And has just been an amazing member of the lab, excellent scientist, hard worker, super contributor to everyone's work and to the efforts of the lab. So I'm really excited to have him here today. So Calvin has his bachelor's from Shanmin University in China and is defending, hopefully, yes, this summer, and then plans after which you can ask him about, we'll see. But he's been working on mangrove encroachment and changing range of mangroves across the Gulf Coast, and we're here about that today. So there you go. Floor is yours.
Thank you, David. All right, so hello everyone. My name is Yi-Young Kong. I go by Calvin here, and as David said, I'm currently a PhD candidate in NRE. So today I'm presenting my doctoral research. It's about the process and impacts of mangrove poleward expansion in the Florida Gulf of Mexico coast. So zooming out for the bigger picture, we know for the past decade we have seen this dramatic increase in global air temperature caused by the accelerating greenhouse gas emission, and that warming is expected to even further exaggerate into the future. And that has caused a lot of different species to change or shift its range with different response, and all of them could have important ecological consequences both globally and locally. Now, specifically in the tropical to temperate transitional zone, we're seeing a large number of tropical species that are moving forward due to the warming winter. So basically, the winters used to hold them back. Right now, as the globe gets warmer, they're able to move toward the pole. That means towards the north in the northern hemisphere and towards the south in the southern hemisphere. One of such species in very critical in Florida is mangroves. So mangroves are the foundational species for coastal wetlands of Florida, and it provides a lot of different ecosystem services like carbon sequestration, wildlife habitat, coastal protection, and so does the salt marsh in the north. So with mangrove being the tropical species moving forward, moving north here, we're seeing them to replacing a lot of those existing salt marsh in the north, and potentially change the ecosystem services and function. So that's why we're studying this issue. Now, specifically, we use the Florida West Coast here to as a study system for understanding this process impact of mangrove poleward expansion. Now, something about a closer look at this this coastline. We see that it's a very dynamic tropical to temperate transitional zone spanning from the tropical South Florida to the more subtropical and towards the temperate of the panhandle. That also is corresponding to associated with these gradient of vegetation that are adapted to this climate. And for example, here in Waukesha Bay Cedar Key region, we have this picture in 2022 that's almost completely covered by this tall and dense and big black mangrove here. But looking back from the satellite images is not always the case. In this particular island, we see that 30, 40 years before is not as fully covered by mangrove. There's a lot of salt marsh, and the mangrove is not as thick. And this is the case for a lot of those island and land coastal wetlands in this region. Now, moving a little bit north, we see that in 2022, again, this picture in Apalachicola Bay, the Panhandle, Florida, we're seeing that mostly the salt marsh dominated coastal landscape, very flat and short, but some scattered and short mangrove, black and red, start to encroaching in. So it really posed the question that when this warming gets more accelerating into the future, will someday the Panhandle coast will look like something in the right? And if that's the case, there will be a huge landscape-wise transformation, and there's a lot of ecosystem functions, as we talked about, related to such transition. So another closer look at this coastline. So this color here is the minimum air temperature of the past 40 years of this whole region. And basically we're seeing this again from tropical south, warmer to the colder parts. Also associated with the gradient of vegetations. So each of the dots are one of the sites that we have as field study sites. And we have 18 dots along this coastline. As you can see, there will be a very distinct gradient of the vegetation as well. So if we drive a pickup truck from the very south, from the about Sanibel and Sarasota County, we're seeing mostly the dominating red mangroves here, very tall and dense, very mature, and it's quite a signature Florida mangrove ecosystem. But as we're driving north towards the Waukesha Bay Cedar Key region, what we call it former ecotones of the mangroves and marsh, now fully dominated by the black mangroves, almost very tall and dense as well. I'm sorry. And going to the very north of Apalachicola Bay, the Panhandle, mostly salt marsh, but we're seeing the black and red mangroves encroaching in as this process is ongoing. That we had never seen this from historical record before. So to basically lay out these chapters objectives of my study, and we can start from this bigger picture of the climate change. We see the warming winter temperature regime, and leading to a process of mangrove pore expansion, especially vegetation. It has a spatial variation that we see across the study ecosystem here, and also the temporal changes of those mangroves that specifically have a focus on the long-term and short-term expansion aimed at freeze events. Notice also this spatial temporal variation also has this different pattern across ecological levels and different spatial temporal scales. And this process has brought an impact. In this case, we'll focus on the mangrove soil. We'll talk more on that for chapter 4. And of course, we have the spatial gradient across its latitude to study it, and also the temporal dimension of the climate, the vegetation, and the soil profile to help us get a better idea. Now, if you're wondering, the chapter 1 is just introduction we have go through. And of course, there's the linkage between the ecosystems, between the ecological levels and the spatial temporal range that we try to understand. So before we dive into chapter 2, I just want you to know this is just for help me to graduate. And if it's getting too complicated here, you will see what it actually means in chapter 2 and 3 and 4. Now, so chapter 2 is about the spatial temperature gradient and the gradient of vegetation along with it. It has been a publication in last year on Journal of Ecology, if you're interested for more of the details. So what we're trying to do here is to fill a gap of getting the field data of mangrove temperature relationship. So the picture we're seeing here is basically a hypothetical mangrove temperature relationships brought up by researchers, but we need the field data to actually quantify this. And after doing that, we'll be able to understand how mangrove respond to different temperature, and in future warming, what kind of mangrove structure are we expected to see. Now, we're not only doing that, but also quantify that relationship across different ecological levels of individual, population, and community to get a better full picture of this variation, and also get a better mechanistic understanding about why those patterns we're seeing has been scaled up. So the research questions we are specifically targeting here is the first to understand the community level, species composition, and above-ground biomass of mangrove how it vary with minimum air temperature, the TMM here, which are known to be one of the limiting factor for mangrove structure. Secondly, we focused on the species-specific population and individual level, again, with its relationship with temperature. And third, we use that information to help us predict how mangrove ecosystem structures will vary across the Florida coast into the future. Now, remember those 18 sites we have, and in each of those sites, we have basically this field sites study plans that we basically lay out a 100-meter transect and 3 100-meter square circular plots where we quantify the mangrove structure here. And also we applied a plotless approach called synoptical highest tree to basically get more of the individual data here and without adding bias to the plot method. So essentially what we did here is just we measured the trees and took a bunch of soil samples, and you'll see how that went. Now, here's how the results are going to lay out. The basic idea, again, is to have those field-based mangrove temperature relationships here across different ecological levels, using that to project onto space under different warming scenarios and see how Florida coasts will look like into the future. Now let's dive into the first one. So this is on the community level. We're seeing the percent coverage of mangrove on the left and canopy height on the right, and how it varies with T-min. Again, T-min is across this latitudinal gradient. So what we're basically seeing is a threshold response of both ecosystem structures here. So basically meaning there has been a temperature range that will brought up the most dramatic response of mangrove as their biological threshold. So also threshold zone is kind of a narrow in terms of the canopy height, meaning that they have a more drastic response to the temperature. Down to the population level, we're seeing that different species here really have a distinct different effect. So basically, there are 3 species of the mangroves in Florida: the black mangrove, red mangrove, and white mangrove, as you can see in the color. And they have very distinct relationship with temperature. We're seeing the percent abundance on the left and percent coverage on the right. Very similar quantification here. So we're seeing that the red mangroves on the right is basically have its dominance on the very warm tropical cells, as we talked about as the virtual field trip. And also moving north, we're seeing the dominance given to the black mangroves as it's more freeze tolerant. We'll talk more about that on the next chapter. And again, to the very left, where it's very cold in the Apalachicola Bay, the dominance is given to the salt marsh. So why we care about the species specific relationship here is because, as you can see in the picture, the red mangroves has this signature prop roots that are very dense and very complex, makes you very hard to walk in those ecosystems. But the black mangrove is quite different, has multi-stem, but it's mostly very straightforward, very tall and dense. And the black mangrove structure also has a difference, vary with temperature. But basically, those landscapes are dramatically different and could have given different preference for different wildlife as habitat, and also provide different coastal habitat, coastal protection ability. So using that information, again, we're trying to project onto the Florida coastline and see how different warming scenarios, in this case RCP 4.5 which is the moderate warming, and the extreme warming scenarios here. So common message across different, we're not going to each one of the detail, but the common message is really the dramatic increase of mangrove structures, especially the coverage and canopy height. As we see, they have a threshold response. Once that threshold has been surpassed by the warming, it will have a very dramatic increase, especially in the northern part of Florida. We're expected to see some dramatic increase. And also the shifting of species dominance across the range, meaning that they will have a very different landscape, even though they're all mangroves. Jumping to the conclusion here, the take home basically, we're seeing the mangrove structure is really temperature driven across latitude, the latitude gradient of Florida, and across the different ecological levels of community population. Individual, we don't have too much time to talk about here, but we're seeing that information carry forward to the warming scenarios where mangroves may further expand and develop in the Florida coastline, especially the northwest Florida. I have the potential of homogenizing the Florida coastal wetlands into tall mangroves. And for more details, please refer to this journal articles. So jumping on to the third chapter, which is have a more focus on the temporal change, and especially one freeze event and how does mangrove respond to that. So again, with a more temporal focus, this one has been a very recent publication as well on the Journal of Ecology. And what we're trying to do here is to understand the freeze response of mangrove. So why we care about the freeze. So as we talked about the Tmin, the minimum air temperature, and we're seeing the freeze event is really one such thing that has been these limiting factors for mangroves, and not only that, but a lot of other tropical species, because freeze events are holding them back. Now with warming, we expect the freeze event to shrink in terms of intensity and also frequency, so that expansion of mangrove could be expected. So understand the linkage between the interaction in between will give us information of how we can project into the future, especially in terms of temporal changes. Now, again, it's a very similar site map here, but we're focused on revisiting a lot of the sites in the north part where this particular freeze event in 2022 has caused impact. So, and that event is color-coded by, again, the minimum air temperature in 2022 here. Now, the research question here is going to focus on, again, species specific, but the low temperature threshold for mangrove resistance and resilience. We'll talk more about that. And secondly, how does the mangrove resistance and resilience vary by different temperatures, species, and stratum to give us a mechanistic understanding of mangrove's freeze response. And third, we use that information again to project, but in terms of not in terms of spatial variation, but more of temporal changes. So building on the last chapter's method, we have this field size, the circular plot, blah, blah, blah. But again, we're measuring something different. We're measuring the basically leaf damage, height reductions, things that could be impacted by the freeze event in those mangroves. And again, we went out 1 year after the freeze event to measure the mortality, resprouts, and basically resprout stems and resprout heights. So this will give us information of how mangrove responds in terms of its damage and how we resist that damage, which is a resistance, and also in terms of how mangrove recover from the freeze event, which is how resilient those mangrove is. So let's again take a virtual field trip. So after this 2022 winter storm, if you guys were in Florida, you probably remember that is during the Christmas time. It's really cold Christmas, and the mangroves are not having a good time by then. So basically, we went out just 1 month after that event and to see how the mangrove responds. And now this is a picture in Cedar Key. We're seeing that 3 mangrove species align well here, and basically very distinct freeze response. As you can see, the black mangrove basically have no damage at all. The white mangrove is taking basically fully damaged with all the leaves turning around in one single site. And also moving on to another site, we're seeing that in one single species of red mangroves, the tall stratum and short stratum are really having a huge difference as well. The tall stratum, the leaves are all gone. The short stratum basically largely preserved. So it's speaking out to some kind of mechanistic of how different species of mangrove can be mediated by the freeze event. We'll see that more with data. And of course, the freeze temperature playing a very good role here with different freeze temperature in different sites. Now, again, after 1 year of the recovery from this particular freeze event, we went out to see how the mangrove recovered, basically their freeze resilience. Now, so what we see is the black mangroves are being the most freeze resilient, meaning that they have the largest number of resprouted branches. We count at least 50 of those branches in one single tree after 1 year. They're very impressive, and some of those branches also happens in white mangrove. So very surprisingly, if there's any mangrove nerd out there, red mangroves are not known to have those resprout branches, but we see one individual with one resprout that's very surprising and very interesting. And of course, they also a lot of them also flower and produce propagate, which is again quite surprising to me. So to speak out how resilient that they really are in terms of recovering from the freeze, and that really speaks out to the investment into recovery, meaning that if they resprout, they're investing in the current generation. They're saying that I want to live. And if they invest more into the flowering and propagate, they're investing more into the next generation. They're saying that, you know, I might not live so long, but I'm going to put all the energy into the next generation, and you guys carry my gene. So basically, we quantify those things we see in the pictures, and it's very similar idea. So we see the low temperature threshold being the lowest in the black mangrove, followed by the red and white, meaning that the black mangrove having the highest freeze resistance to resist the biggest, the most intense freeze. Yeah, sorry. So for the first row here, we're seeing the leaf damage in terms of low temperature threshold, and similar story in the mortality rate. Now, if we're moving on to the plot study and we're trying to understand how stratum has that similar effect. Now, very similar quantification, leaf damage and mortality rate, but in this case, the species of red and black mangroves, but also different stratum of it. So what we're seeing that is the tall red mangroves notably has the highest mortality rate, highest leaf damage, meaning they're taking the most damage. However, that's not the case for the short red mangrove. The short red mangroves almost have very similar response to like the more freeze tolerant black mangrove species, meaning that it's speaking out to how the red mangroves want to preserve their population by, in some sense, sort of the tall red mangroves getting sacrificed, but the short red mangroves are getting preserved due to their proximity to the ground, and the temperatures are relatively higher with some preservation from the top. So again, with resprout rate, speaking out through freeze resilience, we're seeing tall black mangroves having the highest of them, meaning that almost 20%, 15 to 20% of those black mangroves are be able to resprout back and basically live, continue to live. Carrying forward those information into the projection, again, we're trying to see how the temporal change of the mangrove species into the future. Now we choose two sites of interest. One is the current range limit of the mangroves and salt marsh in the very north of Laguncularia Bay here, and also the formal ecotone Cedar Key, where we know already being covered by mangroves right now. So the three dashed line are the temperature threshold. We just quantified the leaf damage temperature threshold, and the other colors are basically the temperature in the past, that's the green part, and also in the future, that's the red as extreme warming and yellow as moderate warming. So basically, a common message here, we're seeing that all those temperatures into the future are surpassing those temperature threshold. Another visualization of that is using freeze degree days, basically meaning that how each of the species of the mangrove feel the cold, how much cold they feel, right? With a different temperature threshold. So basically, again, very distinct decrease towards the end of this century. We're seeing that 2080 or 21,000, that basically means that it doesn't feel much of the cold towards the end of the century with this warming going on. And they could potentially have being lifted by those limiting factors of the freeze event. Now notice how the magnitude of the freeze degree days, how much cold they can feel, again, really resembles a chronosequence, a temperature sequence, a time sequence from the north in about 2060 to a little bit south in Cedar Key about 2020. So that means that, again, bringing back this question we raised before, are those mangroves tall and dense black mangrove forest in the Cedar Key region we're seeing right now be someday to transform into the current ridge limit of Apalachicola Bay. And if that's the case, again, there will be a huge transformation of ecosystem services, and a lot of things are on the line. So to conclude this study, we have this diagram, which is basically a hypothetical diagram to see if a freeze degree can cause different species to respond differently. But the basic idea is that after damaging freeze event, those three species, tall and short, will have different response. And after recovery of the freeze event, it really, again, recover into something very different. And that really explains what we're seeing in the first chapter, that the black mangrove have this, because it's more freeze tolerant, it really has a larger dominance in those freeze mediated regions, which are the ridge limits we're interested in. Also followed by the red mangroves, largely preserved with the short trees, and also the white mangrove being the least freeze resistant and resilient. Again, these are backed by the data, but we already see that, so no need to dive into the details. Conclusion here, the take home is basically we see that the mangrove freeze response really vary by those species at different strata and also the temperature. And we see exactly how and with that quantification. Now projected warming will exceed those mangrove low temperature threshold in those range limits, and especially in Northwest Florida. So basically, we're seeing that those mangroves won't feel the cold and they will be free to develop and expand into the future. So again, very common message combined with the last chapter, the controlling role of minimum air temperature for mangroves structures, especially across the latitudes in Florida. And also the mangroves are expected to further expand and grow in ridge limits. If that's the take home, then that will probably suffice. So again, for more details, please refer to this publication. Yeah, so we see how those process plays out for the mangroves, and we see that expected to further expand in Florida, and what kind of impact it really brought, right? So in this case, we're really interested in the mangrove soil, and here's why. So again, we're chapter 4 is about impact on soil organic carbon. And this has been a submitted manuscript just recently in the spring break, so we'll see how that goes. But so why we care about the soil, all right? So we know that the blue carbon ecosystems, known as the seagrass, the salt marsh, and mangroves, are really the best at mitigating those carbon emissions, meaning that they serve as almost like a carbon pump. They're really highly productive to sequester atmospheric CO2, and they're really good at putting it down to the soil because they have a low decomposition rate. Now, so that's what we see as the data showing that most of those organic carbons are really stored in the soil of those blue carbon ecosystems, not only for the salt marsh and mangroves. So with that mangrove replacing those salt marsh, we're really interested in how that impact is a large carbon storage down in the soil. So again, we use a very similar study sites, 15 sites in this case. And again, we're seeing the variation in terms of the vegetation. But here are the questions of how those soil cores really varies in terms of organic carbon. How much carbon can they store, and what kind of impact does mangrove expansion brought to them? So again, building all this very existing field study sites, we have those vegetation quantified already. Focused on the soil cores, we took three 20-centimeter soil cores in each of the plots, and we combined those depth intervals into the depth interval positive sample, and to send it out to the lab and measure a bunch of stuff we'll see. So again, the research question is what kind of impact does the mangrove expansion brought to the soil? Oh, sorry, one small thing is that in here we sort of frame this whole gradient as a temperature, as a time sequence of how mangrove expands. For example, in the tropical south, we're seeing most of those mangroves being very mature, has been there sitting on decades and even century. And going north, we're seeing that as formal ecotones where mangroves have only been largely replacing the saltmarsh with about 3 to 4 decades. And to the very north of Laguncularia Bay, it is about current ecotones is about 10 to less than 2 decades. So basically, the mangrove exert very different timing of those impact onto the soil. So here's what we're gonna see. We use the soil organic carbon density as a quantification for how much carbon the soil can store. And in this case, we see the SOC density really increase with the mangrove above ground biomass across its latitudes, and also increase with the mangrove stage, right? So meaning that the mature mangroves and formal ecotones with longer mangrove stand, we're seeing that they have a higher ability to store more of the organic carbon in the soil. And the current ecotones, the younger mangrove, at least. However, it seems very tempting to say, okay, mangrove really has positive impact on the carbon storage, but we're seeing that this regional gradient of sites really brought a lot of other factors into the story. One significant factor is a potentially significant factor is that the sedimentary setting, and what this means is that in the northern part of Florida, we're seeing that a lot of sediment coming from the Apalachicola River and whether from the Appalachian Mountain. So basically, you can think of it as diluting a lot of the organic carbon in the soil in the north. That is the younger mangrove stand. And also in the southern mangrove stand, it has been a carbonate sediment setting, meaning that the mangrove are able to sit down onto a very stable setting and accumulate most of its organic carbon without any dilution. So that could really play a confounding factor here. Now, so we further seek out how in each of those sites, those depth changes, those OC density changes across different depths. Now, again, we're seeing OC density, but now in this case, across different depths, in terms of the 3 sites of the mangrove stages. We're seeing a common pattern that there has been increasing of the SOC going to the shallower soil, meaning that it seems like the recent decades of the mangrove expansion may have a positive impact. However, we seek further evidence from identifier of the source. In this case, we use a stable isotope of D13C here to identify the OC source. Where does that increase coming from? So this D13C really separates the C4 plants, which is a lot of dominant marsh of Spartina testiculus, and also C3 plants, which are all the mangroves, but also the Juncus marsh and potentially other saltmarsh here. The patterns we're seeing across depths, across different stages, are that we're seeing no pattern. We're seeing no such increase. There has been a discrepancy between the quantity of the organic carbon and the sources of organic carbon. So meaning that is there not enough evidence to say that increase of organic carbon really come from the mangrove? It might not, and it might be a confounding factor from Juncus marsh or other external inputs. So conclusions here again, we're seeing that younger mangrove stand has the lower SOC density and the higher D13C. The older stands suggesting that there has been lower of the mangrove SOC inputs. However, the SOC does not only vary with mangrove biomass, mangrove expansion stage, it also vary with the sedimentary setting, which are other factors could play a role here. Lastly, we've seen that SOC density, again, across different mangrove stages, across depths, really decrease with that, but D13 does not. So underscoring, again, the common message here is underscoring the impact of a lot of different environmental factors to potentially warn more of the cautions in terms of interpreting the results. So take-home messages, which will probably help you guys for finishing up with this exam, this homework. So the first chapter, we're seeing the structures of the mangrove community, population, and individual. Again, vary with this winter temperature gradient in Florida. And in the second chapter, we see that the mangrove community is really mediated by these freeze events, with different response across different species, strata, and temperature. And we're using that to predict the further expansion and development of mangrove in the warming future of Florida. Lastly, we see that mangrove expansion, this process really has a huge impact, could have a huge impact on SOC, but we see that regional variations of sedimentary setting, other environmental factors really put cautions in terms of how we interpreting the results. Now, what's next? What's next with this? So chapter 5, I promised the last chapter will be short, and yeah. So I'm really working on this, but I'm really excited about this because this can really brought a lot of our field sites towards the next level. So this chapter is about remote sensing, and so why we need remote sensing is because this is a picture of me. Thank you, AJ, for taking the picture, and it's me in the field, and really with me in the field, there's only that much of the spatial temporal range I can cover, and we can take snapshots of the forest community and maybe the cover type, but really never depict a full picture. So to upscale our understanding, to have the full understanding, the full picture of the forest community, the higher level information, we really need something else. And luckily we have that as a satellite, has been around our globe for decades there. So this one, particularly the Landsat satellite by USGS. Now, there has been a lot of open source satellite images data, and they all have different resolution, bandwidths, and temporal range they cover. So what's most relevant here is what we're using is the Landsat 5, Landsat 8, and Sentinel-2 data here. So Landsat 5 and 8 have this 30-meter spatial resolution, and Sentinel-2 has a 10-meter resolution, but less of the temporal time frame that it covers. The research question that we're allowed to answer with those tools are first, what is the timing of those mangrove range expansion in terms of the long term of the starting time and the rate of that? And also, secondly, in a short-term sense, how does mangrove really respond to the disturbance such as freeze event or hurricanes happening in recent years? So when I was trying to gather those data, found there's a gap in terms of how you can retrieving those pixel-based most remotely sensed vegetation index, which is a quantification of how green it is basically, and especially from those data sources. So what I did is I built a website in terms using the Google Earth engine. Here's how it looks. I call it time series retriever. But basically, this apps allow you to draw a region of interest, draw a polygon, specify a data source, and also the start time, the end time of it, and also customize some kind of a threshold for cloud cover, how you want your image, and how much of the image you want to cover the percent of the pixels inside of the region of interest. So all of that, but basically it allows you to have a very general look at first of how the temporal trend of, for example, NDVI and DMI changes across time. And then, more most importantly, you can download this CSV, which are the dates, latitude, longitude, the pixel-based band informations retrieved for you. And this is a format that is very easily going to be transformed into Python, R, whatever tools you want to use for data analysis. So basically, this will help me to help other ecologists and managers interested in, for example, how a vegetation change across different time in their region of interest, to retrieve this data, retrieve a general image and the pixel-based CSV with ease. But there has been limitations. We can talk about that if you're interested after this talk. So with that, what I'm trying to really do is to select again those very similar sites, but in this case the Tampa Bay, Cockroach Bay as the reference of the mature mangroves, and again the Cedar Keepar as the formal ecotones, and the Apalachicola Bay as the current ecotones. Now, pilot study have shown that they have very different NDVI, those remote sensing image information across time, and we're trying to interpret that. But basically, you can see that the chronosequence, the different timing of those mangroves really expansion really happens at different time. For example, the current ecotones of Laguncularia only have this increase with mangrove expansion around 2010. You know, that was the case in 1980 in Cedar Key, and has not been the case for the mature mangroves. So basically, we're expecting to see more of that in the future. But now I really can end this with acknowledgement. So I really thank Hannah Cycles and a lot of folks who have been helping with the field study, and also the founding by SNRE and NCBS of UF and USGS for this research. And here's the reference, and thank you for listening.
So you got 40 minutes.
Exactly, 40.
Yeah, great. Okay, so we'll take some questions from the room. Questions for Calvin, please. We're not just gonna rely on AJ to ask questions, right? So question, go ahead. Yeah.
It might be kind of silly, but for the, I forget this word, receiver, how long did that take you and why did you like do it the way that you did? I don't know if I just missed that part.
Yeah, so this one.
Yeah.
Yeah, so why I did that is because when I tried to get this data, so basically get something like this, and I just found no easy access, easy way to do it. And you can write all the program and stuff, but you know, it take a lot of time for each individual to do that. So what I did is basically to create this website that everybody can, you know, access, and you can just like, there's no code involved in this interface, and you know, you can basically click a couple buttons and get the time and space if you're interested in, and those informations available.
Gotcha. I missed the part where it wasn't a code.
Yeah.
I feel like it's a lot of time on it. Thank you.
Storms, it seems to me that storms would have a big impact on, especially the slow carbon accumulation.
Yeah, storms like tropical storms and hurricanes and things like that.
Especially.
Yeah.
Future. Maybe that might be the next step.
Yeah, that's also a very interesting like comment and question. So like I think, yeah, like you said, maybe the storm are brought in a lot of deposits and they're really adding up to those mangroves. So yeah, in this study in particular, we have not find a way to detect exactly how the storm make impact on this soil course. But I mean, that's definitely true, and especially in a lot of southern parts.
Aren't there SETs, doesn't Chemicals have data from some of those sites? Like around Richley Bay?
Yeah, some of the sites probably, we know maybe there's one site, one SCT in here. Rupert Bay, that's true, and
And I would
Maybe
guess that there could also be soil removal in some sites too, especially with waves coming in.
Right.
and
Yeah.
pushing
Yeah.
us down.
yeah, that's true.
So did you tried to differentiate the C13 and C14, C3 and C4 plants, sorry, and attribute that to marsh versus mangrove, but did you also find the signature of any like marine phytoplankton and other things that might be brought in from the sea that was accumulating in the organic carbon pool? And
So
could you differentiate it?
yeah, so it's been very hard to discern that. But we're seeing that in this case, we're only showing the D13C. We also measure D15N, which is another stable isotope. But combining them, you know, we propose a different series about the potential increase of organic carbon can really come from not only the mangroves, but also the Juncus marsh, and also potentially from the riverine and a marine influence, something like you said, the microphytes, the other different microbes as well, the algae. So yeah, so in this case, trying to identify the source is really the challenge here, and we have collaborator using different biomarkers in terms of amino acids and iron. And it just turns out to be, again, doing a better job at discerning the source as this study, but again, very challenging to have a definitive answer in this case.
Did your research showed any adaptations of mangroves during the polar temperatures? Any distinct adaptations?
Yeah, that's a great question, because that's one thing I've been thinking about also, but yeah, in my particular study, I don't think I can quantify the adaptation but I know that a lot of other studies, especially genetically related, those studies really quantify that as we're seeing, for example, the black mangrove. Those populations are really having the different genetics in the north and versus in the south, meaning that there has been adaptation in the north that they're more tolerating to those cold events. So that's also happening across this gradient, I imagine.
All right, no further questions. Let's thank Calvin again. Great job. All right, we're getting close to the end of the semester, so I'm caught up with grading your write-ups through week 7, which is Robert Nairn, which was a great seminar, really good write-ups on those. Continue to work on catching up. Next week, we have Dr. Cham O, who's an assistant professor in the Department of Environmental Engineering Sciences here at UF. So he'll be presenting genomic surveillance to track microbial fate and activity in natural and engineered environments. So that'll be in this room again. So please show up. And until then, we'll see you next week. Thanks, folks.